Introducing myself, my name is Megan Mahoney, and I'm the Director of Scientific Affairs and Training Programs at Biokenrex. And I'm very pleased to welcome you to our 9th edition of the Summit Speaker Series that we started in lieu of our face-to-face Summit for Cancer Immunotherapy. This Summit Speaker Series is an opportunity to bring our network researchers together, their collaborators, and their patient partners to tell you a little bit more about the research that they're doing. And so today's session is called Harnessing the Power of Tumor Infiltrating Lymphocytes by Cell Selection and Adoptive Transfer Immunotherapy. And I'd like to start by introducing the speakers in a moment, but even before that, I'd like to start by saying, please stay tuned for our next session, which will be taking place in September 2021. We are taking a little break during the summer months. In the meantime, though, you are welcome, and we actually strongly invite you to check out the recorded sessions at our website, cancersummit.ca, and also stay tuned of upcoming events by following us on social media. So today we have 3 speakers. The first speaker will be Dr. Simon Turcotte, who's an assistant professor at the University of Montreal, and who also works as a surgical oncologist and as a full scientist at the Centre Hôpital d'Université de Montréal, so CHUM. His laboratory is focused on identifying new types of tumor antigens and therapeutically actionable immune checkpoints in colorectal cancer liver metastases. Secondly, we have Dr. Sandy Pelletier, who is the scientific coordinator of the Adoptive Cell Transfer Immunotherapy Program at CHUM, and she's been with the group since 2016. And finally, we have Sylvain Bellegarde, who since 2016 has been a patient partner at the Center of Excellence on Partnership with Patients and the Public. He's also been a patient coordinator and a member of the Executive Council at the Canadian Donation and Transplantation Research Program, among many other groups. And I think, Sylvain, you'll be speaking to some of these activities as well. So the format for today's session will be that each speaker is going to present for approximately 20 minutes, and that leaves us about 30 minutes for Q&A. With that being said, we do invite questions as the speakers are presenting, and basically what that will allow other participants to do is upvote questions that are most popular. But I think with the amount of time that we have, we'll be able to get to most of the questions. If you do have any questions, please don't hesitate to write them in the text box, and we'll get to you. But otherwise, I ask you to please write your questions for the speakers in the Q&A functionality. So with that being said, Simon, I'll hand it over to you.
All right. So let me know if you can see my my PowerPoint.
Yeah, looks great.
Okay, perfect. So it's my pleasure today really to talk to you about our research on this project called Harnessing the Power of TILs by Cell Selection for Adoptive Transfer Immunotherapy. There's a lag here, I think. Okay, so as Megan said, I'm going to be presenting first, and then I'll be happy to answer all questions. And I'm going to give the introduction to TIL and the rationale behind our approach. Sandy is going to present features of the cell product we want to infuse into patients. And then Sylvain is going to present what we're initiating, what patients are initiating to help patients face both the end of standard therapy and enrollment into early cell therapy studies, which are complex and not so well known. This work is supported by Iovance Biotherapeutics, and I have unrelated industrial funding for other work. So as you guys, you folks probably know, cancer immunotherapy is a revolution in the past years. There is really nothing as great as a T lymphocyte able to kill a cancer cell, as you see here. the cancer cell undergoes apoptosis after the T cell recognition. And we really want to harness this capacity of T cells to recognize and kill cancer cell as best as we can. The T lymphocytes can recognize cancer cells because these cells have a messed up genome. That genome is full of mutations and alterations that are translated into proteins that eventually gets to the cancer cell surface as a form of short peptides that contains mutations that can be recognized by T cells. So these types of key antigens are called neoantigens. There are other forms of peptides and other sorts of peptides, I should say, that can be recognized by T cells, those derived from viruses that have initiated cancer in specific organs. But this talk is going to focus a lot on neoantigens. So you've heard of cancer immunotherapy. Before I specifically talk about TIL, I want to position TIL in relationship to the first type of immunotherapy that has been widely used in the clinic thus far. This strategy is based on reinvigorating T cells and their capacity to recognize cancer cells by giving patients some antibodies that can block breaks, inhibitory receptors at the surface of T cells. The problem with this therapy, the main issue is that it can be effective, especially when we target PD-1, but only in a minority of patients who probably have a sufficient anti-tumor immune response that we can reinvigorate. But most patients do not have a sufficient anti-tumor response. So this is where we come with cell therapy as a more powerful approach that is really booming since 2017. As you can see here, this is a representation of immune checkpoint blockade studies with a good increase in the number of studies between 2017 to 2019, studies of all phases. But you can see that cell therapy also between those years has been really been developed and is reaching different stages of development and approval. So cell therapy is basically infusing T cells that can recognize cancer cells into a patient. And there are many forms of T cell therapy, anti-cancer T cell therapy. The one we're most interested in is based on tumor infiltrating T cells. There's no better place in the body to find endogenous T cells that can recognize cancer antigens than directly in a tumor. And this approach is done with the three general steps that are shown here. We have to resect a tumor from the patient. We chop this tumor into small pieces, or we make a cell suspensions with that tumor and grow the infiltrating T cells ex vivo in the incubator up to large numbers that are given back to patient as an anti-cancer transfusion. The patient has to be prepared with a non-myeloablative lymphodepletion chemotherapy that lasts 5 to 7 days, usually. This makes room for the anti-tumor T cells. It provides them with plenty of homeostatic cytokines and makes room for the T cells, after which we most of the time give patients some adjuvant cytokines to promote the proliferation of the infused TILs. So this therapy, adoptive T cell immunotherapy with TILs, has been effective and mostly tested in solid cancer, like immune checkpoint blockade antibody. It targets multiple antigens in the solid tumor, just like when we try to reinvigorate the, the, the T cells with immune checkpoint antibodies. The toxicities are low. They can be due to when some toxicities happen, they're most often due to the lymphodepletion, lymphodepleted states in the form of bacteremia or some infections that can readily be treated with antibiotics. In contrast with blocking antibody, this is a one treatment deal, and this needs to be done at the hospital, in contrast with the blocking antibodies that are given long term, every 3 to 4 weeks, depending on the agents. So TIL therapy thus far has been tested mainly in melanoma. As you can see here, with this is snapshots of most numerous and most recent trials of TIL in melanoma in hundreds of patients. And you can see that the objective response rate has traditionally been between 35-ish to 50% response rate, with some complete response rates reported in as much as 17% of patients, and 2-year survival around 40 to 50% of patients. Of note here, patients that are now receiving TILs have received prior other immunotherapies, and they've not responded to prior lines of many therapies and immunotherapies, and so they're further down lines of treatments and probably have tumors that are not as well recognized by the immune system. And we think that this explains, this is part of the reason why the complete response rates have dropped over time. There has been several proof of concept that this TIL therapy can also work for other type of solid cancer, as cervix, gastrointestinal, and lung cancer, which I'm not showing here. This is an example of a melanoma patient that has had a liver full of metastases. The darker circles in the liver here. This is 1 month prior to TIL therapy. 1 month after TIL therapy, you cannot see many of these dots. And this is an example of a patient that has been cured by this treatment and has no evidence of residual disease 5 years after its treatment. Another snapshot of a success is the proof of concept that this therapy can work in a patient with metastatic GI cancer, in this case, a bile duct cancer patient who had received several lines of cytotoxic chemotherapy, scans with lung metastases prior to TIL, and 2 years after residual disease, which is much smaller than prior to TIL therapy. So the real question we're facing now is how can we increase the complete response rates and translate the efficacy of the TIL approach to common cancer other than melanoma. And we really believe that response rates can be improved by administering a TIL product that is going to be enriched in tumor-reactive T cells. The with only a minority of infiltrating T cells that are really tumor reactive or neoantigen reactive. And the bystander T cells that do not recognize neoantigens or other cancer antigens that have infiltrated the tumor unspecifically just due to inflammation can overgrow the tumor-specific T cells in ex vivo in the incubator when we use the classic approach to produce TILs. This can be illustrated here in an example with 2 melanoma tumors where you have the percentage of neoantigen reactive TILs in this patient and in that patient. You can see that most of the time it's around 1% of the TILs that can recognize a neoantigen. And when you do the classic polyclonal, unselected ex vivo proliferation approach, one does not control the expansion of those tumor-reactive T cells, and one can lose the tumor reactivity, the neoantigen-specific T cells being diluted by other cells. Sometimes the specific cells can grow, but this is very variable from one patient to another. I'm gonna skip this. So we've developed a way to select tumor-reactive T cells, enrich in tumor-reactive T cells based on an expression of a surface protein called PD-1, which comes up to the surface of a T cell that has repeatedly seen its neoantigen or its antigen. And Sandy's gonna, after my presentation, she's gonna describe what she and we have optimized and what are the features, the key features of the cell product that is enriched in tumor-reactive T cells that we plan to infuse to patients. I'm going to close this introduction to TIL and our approach with a short description of the first trial we are working to launch as soon as possible. This is going to be a phase 1b trial for metastatic melanoma patients refractory to anti-PD-1 immunotherapy using the adoptive cell transfer of PD-1 selected autologous TILs that we're going to give with IL-2 after lymphodepletion. In short, it's called the Selected Tumor Infiltrating Lymphocytes Against Refractory Melanoma Trial, the STAR MO1 trial. This is going to be a prospective open label trial. It's investigator initiated by us at the CHUM, sponsored by the CHUM, supported large PD-1 selected TIL product. And we plan to treat patients with melanoma refractory to first line or further lines of standard therapy. We want to submit our clinical trial application by the end of the year, and we think the enrollment is going to take about 30 months. This is a little small probably, but it's a description of the patient journey. Patients with refractory melanoma are going to be recruited and stratified based on their prior response to first-line immunotherapy. They will undergo tumor resection so that we can produce TILs, the selected TILs. After patients recovered from surgery, they are admitted for lymphodepletion. They receive their TILs on day 0. Then IL-2 is given up to 6 doses. Patients stay at the hospital for a good week to recover from the lymphodepletion and cell infusion. And then after a first day 30 visit, the assessment starts at 2 months, every 3 months, with clinical visits and CT scans, up to progression if it occurs, or down to 10 years if possible. The primary objective is going to be safety and feasibility. It's a new cell product. This exact cell product has never been infused into patients. So that's why this is our primary objective. But obviously we want to hopefully have a signal and capture effectiveness of this therapy as a secondary objective, response rates. And there's an elaborated set of exploratory objectives. We want to understand upfront what is associated with responses and resistance to this form of cell therapy in metastatic. can be found within a patient's own tumor, can be expanded to large numbers in an incubator for transfusion as cancer immunotherapy. This form of cancer immunotherapy is called adoptive cell transfer, ACT, of TILs, tumor infiltrating lymphocytes, and has shown good efficacy in some patients with metastatic solid cancers refractory to all known treatments. With a new cell sorter, we are developing a cell manufacturing process using cell selection for enrichment in tumor-reactive T cells, hoping to improve response rates, clinical efficacy in patients. And the first trial is going to be conducted in patients with metastatic skin cancer, refractory to Health Canada by the end of the year. I'm gonna do the acknowledgements right now so that Sandy and Sylvain don't have to do it. This work is highly multidisciplinary. It takes a lot of resources to do cell therapy in an institution, to produce our own cells, etc. And so there are many people involved, which I will not all mention here. We're really thankful to BioCanRx for their support, the Institut du Cancer de Montréal, who has allowed us to acquire the new cell sorter, and the Shim Foundation, who's has been playing a key role for initiating the supporting the first trial. So I will be, I thank you for your attention, and I'm really happy to answer all the questions on TILs and the approach we've taken so far. Thanks.
Thanks, Simon, for that. Would you like questions now? I do see a couple coming in, or would you prefer to wait till the end?
No, I think it would be good to answer questions now and after every presenters, and at the end we can also have a general discussion.
Sounds great. Okay, great. So before I ask the questions coming in through the chat box, just a reminder that if you do have additional questions, you can raise them in the Q&A box, and that allows other people to vote on them. But the two questions that came in are, is there any evidence to support the tumor-reactive TIL marked by PD-1 expression recognize the same PMHC targets in the primary tumor in comparison to metastatic sites? For example, are immunodominant PMHC targets similar in metastatic sites versus primary tumor sites for PD-1 positive TIL selected from primary tumor site?
Right.
So that's the first question. And then
Maybe
maybe
I can answer now.
Sure. Yeah, sounds good.
It's complex. So we're designing this and can test this first cell therapy cell product in the metastatic setting. So our first goal is really to have a TIL product that recognizes the metastatic disease. The question here is whether the cell product can recognize the primary tumor. One has to understand that in most patients we're going to treat, the primary tumor is going to be resected. But to specifically answer your question, I think it really depends on whether this is a mutation that has occurred early in the transformation process, if it's oncogenic derived, if it's a dominant mutation that has been shared to all daughter cells. And we have good examples that dominant TILs reactive to neoantigens that we find in metastases can also recognize some of these mutated genes that were expressed in the original tumor. This is an example in the primary colon cancer in a patient operated in 2010. You can see we've cloned many TILs with different distinct TCRs that are color coded here. When we do TCR sequencing of the primary tumor, the dominant clone recognizing mutated PAM had the highest frequency of about 8% of TILs in that primary tumor. It was also found in the colon relapse of that patient. And then a year later, this patient developed liver metastases, and the dominant clone was still present in that tumor. So it's going to depend from one neoantigen to another, but we certainly have cases where we found neoantigens that were also expressed in the primary tumor that are recognized by TILs.
Thanks, Simone. Very comprehensive answer. The next question is, could combining PD-1 selection with the expression of another checkpoint, such as like 3, be beneficial in further stratifying or selecting tumor-reactive TILs?
Yeah, it's likely that combining cell surface markers beyond PD-1 is going to yield a cell product that is going to be more specific towards neoantigens. Thus far, the published literature supported that PD-1 was capturing all the neoantigen-specific T cells, but it also brings some non-specific T cells. We had to start somewhere to develop this therapy. It's not, as Sandy's going to show, it's not obvious to sort out T cells from a complex cell suspension like a freshly resected tumor. And to expand those cells, one has to start with a decent number as well. So if you start combining many markers, you also restrain the number of cells that you can sort to start with. And so there has been a compromise between feasibility and the scientific rationale to come out with a first study, a proof of concept study, and we decided to really focus on PD-1 as the most feasible and scientifically sound approach.
Thank you. Great, wonderful, and the person thanked you for the comprehensive answers. So Simone, thank you for your talk. And I think at this point we are progressing to Sandy. So welcome, Sandy. I'll turn my mic off and take it away.
All right. Can everyone see my slide?
Yep, looks great.
All right. So here are my disclosures. Okay, so I'll start with the Simon slide that shows that, let me just get this out of the way. All right. That PD-1 is expressed by TILs that have repeatedly seen a neoantigen and can be used to select and expand tumor-reactive T cells. So the first publication that showed this is from 2014. They had sorted PD-1 positive TILs, PD-1 negative TILs, or unsorted total CD8 TILs. And then they had co-cultured these TILs with autologous tumor cell lines. So as we can see on the left, the PD-1 TILs specifically upregulated the 4-1BB marker, which shows that they specifically recognize the autologous tumor, whereas the PD-1 negative or the total CD8s only poorly upregulated the 4-1BB. In the middle graph is the secretion of interferon gamma by these TILs. So we can see that the PD-1 TILs secrete much more interferon gamma than the PD-1 negative or total CD8 T cells. And finally, on the right graph, they show the fold expansion of these cells in a rapid expansion protocol. So we can see that both the PD-1 positive, the negative, and the total CD8s, sorry, my cat is in, she's caught in my microphone. Sorry about that. The joys of working from home. All right, and so Finally, these show that the expansion of the PD-1 positive, negative, and total CD8s is similar between all three populations. All right, so the next study was performed in a mouse model. So they used mouse that were injected with the B16 ova melanoma cell line, and then they isolated the TILs from the tumors. And finally, they sorted PD-1 positive and PD-1 negative, and then co-cultured them again with either B16 ova cells in black, an irrelevant tumor cell line in gray, with culture media in white, or as a positive control, they used anti-CD3. So once again, we can see that the PD-1 positive in black secrete much more interferon gamma in the presence of the B16 melanoma cell line. In panel B, what they did is that they performed adoptive cell transfer with either the total CD8s, the PD-1 negative, or the PD-1 positive TILs. And they show that the mice that had received the PD-1 positive cells in black, that the tumor size was much lower. So as Simon mentioned, our approach is to perform adoptive cell transfer with PD-1 selected TILs. So basically, similar to other ACT studies, there will be tumor excision, and we will transform the tumor into a cell suspension. And then the PD-1 TILs will be selected using the Max Quantito cell sorter. These TILs will be expanded, and finally, they'll be infused back into lymphodepleted patients. So this is a picture of the Tito in our clean room at the CHUM. So the way this cell sorter works is that everything is contained in a sterile disposable cartridge. So you bring your cartridge into the hood, and then you add your cells into a red, blue, violet laser. And then the cells of interest, in our case, the PD-1 positive cells, the valve will open and let these cells through into the middle chamber. And the cells that are not of interest will go into the negative chamber. So just briefly, this is our new clean room at the CHUM. It is a space for the manipulation of biospecimens for clinical use only. And it is within the operating room of the hospital. So the tumors go directly from the OR into our clean room without going into the pathology. There are two culture rooms. One is dedicated for our study. And there's a quarantine space for delivery of new material until it is released by QA/QC. And the number of air particles, they meet standard class ISO 5 for the culture rooms and ISO 6 for the other rooms. So I'll next show you some data that we had generated. So on the left graph is a percentage of PD-1 positive cells gated on CD3, either in melanoma tumors or in gastrointestinal tumors. So as you can see in both types of tumors, the mean is around 40% of PD-1 expression, although it varies considerably from one patient to the other. So this is before the sort. And after the sort, in the middle graph, we can see that we obtain very good purity. So we get on average above around 90–95% of purity. And the viability is always above 97% after sorting. And finally, on the right graph is the number of cells of PD-1 cells that we can obtain from one tumor. So not surprisingly, the number of cells obtained is proportional to the weight of the tumor that we get. In general, the mean number of cells that we can isolate from a tumor is around 500,000 cells. It can go up to a couple of million cells, and in other cases, we've also seen a couple of thousand cells only. So after the isolation of PD-1 cells, our goal is to expand them. So for this, we use the G-Rex flask. So we use a mix of rPMI and M5 medium. We add 5% human serum, anti-CD3, and irradiated PBMC feeder cells, and also IL-2 for 14 days. So as you can see here in the middle graph, on day 0, as I mentioned in the previous slide, we can get as low as a couple thousand cells and as high as a couple million cells. And after 14 days of expansion, we can get up to 10 to the 9, sometimes 10 to the 8 cells. On the right graph is the fold expansion. So on average, we get about, I think there's an error on the graph. It's around 5,000 cells, 5,000 fold expansion, sorry. So the next thing is we were interested in looking at is the phenotype of these TILs after expansion. So we looked at CD62L marker along with CD45RA marker to look at the frequency of naive T cells, central memory, effector memory, and terminally differentiated TILs. So we looked in all 3 populations, so PD-1 positive, PD-1 negative cells, and bulk total TILs. So as you can see, the majority of cells, like in CD4 and also in CD8 T cells, which is the bottom line, are majority central memory and effector memory, and the frequency of terminally differentiated TILs is very low in this population. So finally, we wanted to test if these PD-1 T cells recognized neoantigens or autologous tumor cell lines. So as I just described, the first step is to generate effector cells. So either we generated the PD-1 positive cells, PD-1 negative, and the unsorted bulk TILs. The second step is to generate targets. So we either generated autologous cancer cell lines, and we also performed tumor sequencing to identify neoantigens, and we synthesized peptides for these neoantigens. The third step is a recognition assay. So we co-culture the TILs with either the autologous cancer cell lines, or we co-culture with antigen presenting cells, such as dendritic cells, that are pulsed with the neoantigen peptides that we had previously synthesized. And the readouts are either upregulation of 4-1BB, which is mostly by CD8 T cells, or upregulation of OX40 by CD4 T cells, and also the secretion of interferon gamma. So this is one example in patient 400-109, where we had co-cultured the TILs with 2 autologous cancer cell lines. So the first one is called MEL1091, and the second cell line is MEL1092. So we can see that the TILs specifically upregulated 4-1BB and OX40. So 4-1BB is on the y-axis and OX40 is on the x-axis. We also use the MCF7 cell line as a negative control. So this is a breast cancer cell line, and the anti-CD3 as a positive control. So this is the results for the PD-1 TILs. So next, if we look at the PD-1 negative TILs or the total TILs, we can see that the recognition of the tumor cell lines is much lower in this patient. So it seems to be really specific to the PD-1 positive cells. So the previous slide showed one example in one patient. And this slide is the summary of all 4 metastatic melanoma patients that we have tested up to date. Some of them have only 1 autologous tumor cell line, and other patients have 3 or 4. So in panel A, we can see that the specific upregulation of CD134 by CD4 T cells is much higher in the PD-1 positive. And on the bottom is the specific upregulation of CD137, so 4-1BB, in CD8 T cells. So we can see once again that in most of our melanoma patients, the PD-1 population seems to be enriched in neoantigen, not neoantigen, but in tumor-specific antigens. So the previous slides were TILs co-cultured with autologous tumor cell lines. In this slide are the TILs that are co-cultured with the antigen-presenting cells and the neoantigen peptides. So for the first patient, patient 2399, we had identified 28,000 single nucleotide variants. And then we identified 225 non-synonymous and transcribed single nucleotide variants. We then use best predicted MHC class 1 affinity to finally synthesize 118 peptides. And we identified 4 of them that were recognized by the PD-1 positive TILs, and they were not recognized by the bulk or the PD-1 negative. So here are the results. So we can see that, so the first graph on the left is for 1BB upregulation in PD-1 positive. So we can see that the peptide RUVBL2, the mutated peptide, sorry, is specifically recognized. In the bulk TIL, there is no expression of 4-1BB or interferon gamma secretion. On the bottom is OX40 upregulation in CD4 in another mutated peptide. And once again, the recognition was not specific to the bulk TILs. And here is another patient. So this patient is a metastatic colorectal adenocarcinoma. And this patient, we had identified 18,000 total SNVs, 35 non-synonymous and transcribed. And we have finally synthesized 35 peptides. And we found that 1 of the peptides was recognized specifically by the PD-1 positive population. So we can see here the peptide, the mutated peptide FRYL is specifically recognized by CD8 T cells. So in conclusion, we have acquired a new closed system flow cytometry cell sorter and optimized the selection and expansion of CD3 positive, PD-1 positive TILs from fresh tumors. And after cell sorting, these TILs have a good viability and they have a good expansion capacity. And finally, the PD-1 TILs are enriched in T cells that are capable of recognizing autologous tumor cell lines and neoantigens. So are there any questions at this step?
Thanks, Andy, for that presentation. We do have a few questions coming in. So I'll turn first to the question in the Q&A, which is, what is the recovery percentage after sorting?
The recovery in terms of
I think it's your final product. I think it came in earlier, so I think I would assume that it's the final TIL product. But perhaps if someone wants to elaborate on that response. And then maybe as we're maybe waiting on further clarification for that, the other question that came in was, is there any evidence that PD-1 selected expanded TILs can support multiple rounds, for example, serial killing of target cells in vitro, or other evidence that selection and expansion of PD-1 TIL does not compromise TIL potency?
So I'm sorry, can you repeat the question? Is it about multiple rounds of expansion?
Yeah, sorry, Sandy, I talked so quickly. Let me rephrase this. So is there any evidence that PD-1 selected TIL, expanded TIL, can support multiple rounds multiple round serial killing of target cells in vitro, or other evidence that the selection and expansion of PD-1 TIL does not compromise TIL potency.
So can I answer, Cindy? Or?
Go ahead.
So we've never tested, it's a good question. We've never tested, made several rounds of killing assays, of cancer cell killing assays. Most of the time, the cell product is tested as a one-shot deal for tumor or neoantigen recognition, but not maintained in culture and retested. I think that's the question here is it relates to the stemness or the capacity of those cells of self-renewal and further proliferation. We haven't done specifically those assays. In terms of PD-1 selected TIL function, the proliferation, as Sandy showed, is quite impressive, and the interferon gamma capacity, and we have some killing assay data, is good. It's variable from one neoantigen specific TIL to another, from one tumor to another, but the results we generated and the data published both in vitro and in vivo favors that PD-1 in this context is a marker of a T cell that has repeatedly seen its antigen, and not just a marker of exhausted T cells with no capacity to secrete interferon gamma and or kill the target.
Okay, great. And lots of questions coming in. So we have a nice amount of time, so I'll move on to the next question. Let me just make sure that I'm starting at the top here. Okay, so the next question is, is there any evidence to support the tumor-reactive TIL marked by, actually, I'm sorry, did I ask this question already? I think I did. Sorry, the question is, could combining PD-1 selection with the expression of another checkpoint? Oh, I did that one too. Sorry, folks. If I could ask folks to put them in the Q&A box. The chat box is getting a little bit chaotic here, but here we go. Sorry about this. Okay, so at the end of expansion, what does the profile of exhaustion markers, for example, co-expression of multiple immune checkpoints, look like on the expanded TIL?
We did study that. We did look at a couple of markers. If I remember correctly, expression of these markers were similar between the PD-1 negative and PD-1 positive populations. It's been a while. Simon, do you remember exactly what the data looked like?
There's definitely more PD-1 expressed at the surface of the PD-1 selected TIL as expected. And co-expression with other marker, which is a feature of neoantigen-specific T cell, is also more frequent in the PD-1 selected TIL product compared to bulk expansion unselected or PD-1 neg fraction expanded.
Thank you. Okay, and so going back to that first question regarding the recovery percentage, it was regarding the PD-1 positive cells. I know you spoke to it. I know it was quite impressive, but did you want to elaborate on your answer to that question?
The recovery after cell sorting?
Yeah, it's after cell sorting. I can see in the Q&A, which you showed.
So the viability?
The recovery percentage.
The recovery percentage.
If you go back in the slides with the pre and post sort.
Can I go back? Oh yeah, there we go.
Right there.
This one. So after sorting, yeah, the viability is very high. It's always above a 97% viability after sorting. So the sorting seems to be very gentle and does not seem to affect the cell viability. I hope this answers the question.
And then next, we've got 3 more questions, and I think then we'll check for timing. But the next question is, how are tumors digested prior to sorting? Are there GMP enzymes for this?
So we do a mix of enzyme and mechanical digestion. So we use the GentleMax system for the mechanical part. And as far as enzymes, we use there's a new GMP collagenase available. And we also use the DNase, that's a clinical grade enzyme. So we've had no issue with this, with Health Canada during our pre-CTA meeting.
Thank you. Okay, and have you tested the isolated TILs versus CAR T cells generated against the neoantigens you identified?
No, we haven't.
And rapid fire questions here. Would there be any advantage for transfusing cells into patients that are both PD positive as well as, for instance, CD137 positive or 134 positive?
You mean, I don't think I understand the question.
Co-expressing in patients that are co-expressing or expressing both PD1 positive and 137 or 134 positive.
Mm-hmm.
And sorry,
CD. OX40.
Yeah. So we haven't looked into that, but we know that there was a clinical trial at the NIH that did sort the 401BB positive CD8 T cells and injected these cells into the patient. I don't think the study is published yet, but they did get overall good response rates, but we haven't looked at co-expression of sorting the co-expressed cells.
And I think that goes back to one of the questions earlier, right? It's just, it's a matter of prioritizing and moving forward with a rational scientific strategy, I think, to what you alluded to, Simo.
Mm-hmm.
Yeah, and we use 401BB and OX40 as markers of de novo engagement of the TCR with an antigen. And so when you take a fresh tumor, those markers are expressed at various level, but when you sort based on 401BB, for instance, you're sorting a cell that has recently seen its antigen. PD1 has the advantage of showing you a cell that has repeatedly seen its antigen, whatever the timing was. So it's a minority of cells will express in the fresh tumor 401BB, and this trial at the NIH that Sandy alluded to, that hopefully is going to be published soon, had technical difficulties in sorting such a small fraction of 401BB cells from the fresh tumor. And so we opted for the PD1 positive, which captures 401BB positive or negative cells as well.
Okay, one more question before we move on, and that is, to what extent did the expanded TILs remain active against cancer in the immunosuppressive tumor microenvironment?
It's an excellent question. Simon, want to take it?
Yeah.
Go ahead.
Well, there's no straight answer to that, right? What is underlying the question is that the immune suppressive ligands like PD-L1 is going to be still expressed in the tumor microenvironment, and it's true for any sort of T cell therapy, and what this question leads to is whether we should give TILs combined with anti-PD-1 therapy as to avoid our TIL product being exposed to the immunosuppressive PD-L1 ligand. There certainly are some examples of patients who were refractory to immune checkpoint blockade of PD-1 who received TILs and then received again anti-PD-1 and responded to anti-PD-1 because after TIL their T-cell repertoire was modified, a little bit like a small bone marrow transplant, sort of. And these are strategies we're thinking about. Piggyback trial, testing the efficacy of anti-PD-1 after our TIL product has been infused. But as of yet, the priority is really to test as soon as possible the safety and our capacity to make the cell product. And then we'll, in a next step, we'll, well, during the trial, there's going to be a lot of correlative work to answer your question. And in a future approach, I think we're going to have to combine this therapy with some immune checkpoints.
Wonderful. Well, thank you. Those are some really excellent questions. So thanks to the participants who are actively participating as well. Sandy, thanks for your presentation. And I always like to see a cat or two show up on virtual events. So thank your little friend there for being involved in the sciences as well. So we'll stop there for questions. And I think we'll turn next to Sylvain to present. And then I've got some questions just about your process and maybe some lessons learned on GMP and how you approach this project, and it's quite a comprehensive series of activities that you've undertaken. So I look forward to the next Q&A. But for now, Sylvain, I'll pass it over to you.
Okay.
Thanks, everyone.
Hello, everyone. So can I share my screen? Yes. Merci. Yes. So thanks, everyone. Thank you. Sandy, Simon, and Megan to give me the chance to be with you today and present on patient partnership in that great project. Is everyone with me in the presentation? Is it showing up? It should be.
Yeah, looks good.
Oh,
yep.
Okay, thank you. So I want to point out, first of all, so as you said, I work at the Center of Excellence on partnership with patient and the public. It helps me to have it in English because I'm used to see it in French. Just a short thing about what we do so we're based at the Montreal CHUM. We used to be at the CR CHUM, but we're now holding our office and being managed by the CHUM. So we're basically a group that works and support patient partnership in research, but also in clinician improvement, in clinic improvement, and lots of things. So I'm there because I've been assigned by Antoine Boivin, Dr. Boivin, as a manager, project manager for the great project of Simon and Sandy. And basically the CEPPP is doing a partnership in education. So we have a lot of project going on in education. We also have partnership in care. And when I say that, it's quality improvement, governance, health, and provincial and Quebec and also national, and we have Accreditation Canada and all that. And we also have, so I was a patient coordinator in multiple, and I'm still in multiple network, research network, like the Canadian Donation Transplant Research Program, CANET, and now on other projects. So it's a lot of things that are going on at the CEPPP. I was listening to Simon and Sandy, how fascinating, but how ununderstandable sometimes the language is in research. And it seems to me that even for cancer or any disease, there's always a different point of view for wherever you're looking at, and what is your specialty in doing it. And I feel when I listened to Simon and Megan, I said, well, maybe I'm the imposter in there. And but I wanted to take you really quickly to my living experience. So why am I here today? So my journey started in 1980, where my sister died. She was 18, I was 13 at the time. You'll see why I'm saying that. And she died from a heart attack, and I was diagnosed with hypertrophic cardiomyopathy at that time. So that was in 1980. The first visit that I had with a cardiologist at that time, I remember he said, well, the only way of saving you would be to get a heart transplant. But right now in 1980 in Canada, we're not doing any heart transplant. So the program was barely starting. So I've been testing, I've been guinea pig for years. But in all those years, I got finally 2 heart transplants. I was the 3rd in Canada in the early 90s to have a defibrillator, the pacemaker combo. And right now, I also live in the last month. So I had 2 heart transplants. One was in 2000, the last was in 2015. And it's so far it's good. I could say on clinical trials, I've been part of lots of clinical trials. One was on my first heart transplant, a couple of days before my first heart transplant, because I was in the chronic heart failure, I would say palliative and end-of-life heart failure, and it started to be really bad. So they actually proposed me to be on dobutamine, was a medicine for improvement, surviving heartbeat, and then it wasn't sufficient enough. So I said, well, we have a new cell, a new cell that could work. So let's put you on Melrinon and we'll see how it works. So I was testing the efficacy of that. And when I listen and when you come to the project that we're coming in, it's always a different point of view from a patient about the risk and benefit. Most of the science is based on 0 risk or at low risk. But when you tell me, well, the week is rough, Sylvain, and we don't know if you're going to last to the end of the week, and you offer me a therapy that could save me at 30%, well, I think it's fair enough for me. So the risk and benefit, that's different from the scientific point and the patient point, sometimes depending on where they are in the planning of life. And this is an important thing to put on. Lately, in the last 18 months, I've been put on, I have skin cancer diagnosed with the Lynch syndrome, which just confirms why all my cousins and aunt on the side of my mom just died all from cancer. So for me, participating in that project, it's just like suddenly realizing that it's just common sense that I might be collaborating in something that I might be, have advantage of participating in it. And just in my family, 2 out of 3 kids had cancer. So the cancer is really strong in my family. But if I go, let's go now to the basic of that. And this is the Montreal model that we see and the patient partnership. Patient partnership, and I should start maybe by saying patient. I also want to mean caregivers. And patient partner, we mean patient partners as people that are involved in a research as collaborators. So there's a big difference in being patient partner and being a patient subject. So some people, I had once one researcher who said, well, the patient partnership part of my research is that when the patient will be on a treadmill testing his electrocardiogram, then I'll talk to him. So this is the partnership part. This is not really it. And we actually, patient partners is a collaborative recognition of both experience. I'm a patient. I did not understand everything that Simon and Sandy just said, and it's okay. I don't need to understand everything. I just need to understand the major part of it. And my job is not to tell them what to do. It's to see what I can bring to the benefit of their project. If you ask a patient, then he would tell you. If you ask one subject from the research project, if you ask them prior or after the research project, then they will tell you how it was for them. What was the effect, the side effects, how it was. So I like to say sometimes you know why, the researchers and the clinicians know why, and the patient know how. So I could tell you lots of things, lots of anecdotes about how I understand my pills, what are the effects, how I manage myself, my medication and myself to make sure that everything is going good with being a 2-time heart transplant recipient and taking 25 pills a day. There you go. So I will skip that one. I just want to point out that patient engagement can be everywhere. And it could be in information, consultation, collaboration, and partnership, just like we are basically doing in there. And to have patient partners in a project, you don't take the first, if you want to do patient partnership, you don't go out of your office and in the hallway, and then you just take the first patient that has an IV pole in your hallway, and you say, you'll be my patient partner. You have to choose carefully, just as any collaborator, your patient, to make sure that first he knows he knows the disease, so he has a related experience with cancer or the subjects of your research. He's able to mobilize his knowledge. He's resilient, especially in a project like Simon and Megan. This project is having a resilience and having the right to give back, and also being able to reflect on his past experience and what is in front of him, and being able to connect and listening to what we are. Why we want to listen? Because this great project, so Simon has approached Antoine, and now we are in it. And the objective of that project is that CHUM and patient public partner in research will define a strategy to create a support group from and for patient enrolled in that current trial, aiming to facilitate informed, accurate, and to provide emotional and knowledge support. Having been in a kind of end of life situation, there's a lot of things that is happening in your head. And as I said, there's a lot of things that are really clinical and the challenge, but also there's a parallel part of that being in a clinical trial, end of life clinical trial. And it's really also a lot of emotion. So one of our goals in that is, first of all, maybe first to help for the consent. We all know that the last time that I have a consent form on a research project, I think it was 21 page long, and I didn't have the force and the brain to understand it. So can we make a consent form more understandable? Then I look at T cells, B cells, Tregs, all the words that were said. Can we also make it easy so the subjects that will accept to participate in that project will at least understand the basic of it and why they get in it? And one of the most, and the support group is, first of all, you get in an endeavor, embarking in an endeavor. This is when you are proposed a end of life clinical trial, let me call it like that. And Simon, you correct me if it's not. It's a decision. And it's usually also a family decision and a personal decision. It's either you go in or you don't. And for me, it's really important. And it's also important to be able to share, but also with the group that we want to go. It's also creating a safe space for that. So the subjects will have lots of experience to go through. They will discover new things with the clinical team also, but also it's great also to have patient partners that will create that safe space of what is the experience, what is the knowledge that I need to have, but also being to share how it's lived in this clinical trial and how they live it. Partnership will be also really important with the clinician and the researchers, because it will be it will be not just a silo. So it's not just like Simon is doing his things and all that. So we want to have the patient partners will be probably the link in all the, more or less in the all departments. So to help support the patient, but also to be a link with the clinician and the researchers, and also to empower. One short anecdote: when I was waiting for my heart transplant, and by the way, the night before, they told me, well, Sylvain, it's looking pretty bad. So if tomorrow we don't get a heart transplant, we don't get a heart for you, you will get on the Jarvik, which was the artificial heart at the time. That was, it was a hard time, but the best support that I got was from another patient that was across my hallway and came to my room and talked to me. And we talked about all the challenges, being afraid of what would be the treatment, being afraid that Jarvik wouldn't be good and things like that. So it's the empowerment of each other from patient and from people that know the experience is also important. So collaboration will be a key in this project. And I wanted to bring in patient partnership. There's there are winning conditions for sure. And I have to, I have to put that thing for sure. So I think the common understanding of the role and responsibility for clinicians and all participants in the research, also working together to encourage and support activities for the patient. So this is really important. And I think in patient partnership and collaboration, we really think about also integrating partners from the beginning and even in the design. So I'm not saying a lot of things because we're starting to recruit. Actually, Simon, I have good news. I have one patient right now, two coming in maybe, one patient partner, but we will start to design the platform and support group and all that with patient, with the feedbacks from what they are. And also from there, then we get regular meeting. And I mean, regular meeting also with transparency. So it's not just research, it's also all together we're doing it. And don't be afraid to overwhelm the patient partners. Keep them informed. And if it's too much info, they will tell you. But don't keep them aside just when they need you. You might be surprised how continuous support could be good. And the expert, the patient will have this expertise also to bring in as collaborator. You know why, we know how, but we know how to get also empowerment and a good recruitment for start, but also the good practice and learning together. This project is really special, special for me also, because but it's really very important. And the Simon reaching for us on that experience, I'm really looking forward to see how this is going to end and how this is going to look through the journey of the subjects, and see if we can improve at least their quality of life or emotional life during this research. This is what I wanted to say. Voila. And I will stop sharing. Voila.
Sylvain, thank you for sharing that. And I think we can anticipate where you'll be tonight. At what time is it? 8 o'clock when the first game of the finals is on? By
the way, I don't like that much hockey. I just put it
really,
because it's trendy. Sorry.
It's very well. It's very relevant, obviously. But thank you for sharing that basically your story and your motivation is very clear, and it's really it's very, I think, motivating. So to participants, I do invite you to ask questions. And in the meantime, I thought we could go back to what you started with, and that's the idea of the imposter syndrome. And I mean, I think that's a very real and important kind of framework to work from. So I think my first question for you is how and what advice would you give other patients or patient caregivers who may wish to become an advocate and they just don't know where to start?
And for me, there's so you're bringing the advocate, and for us at Montreal, there's a big difference between an advocate of defending the role and
Mm-hmm.
the rights for the patient, and supporting and being constructive on bringing an added value to that project. So, it's first of all being able, to be able to manage your own health. So someone that has an issue with health and doesn't care and is not as an advocate on his own health, so he doesn't care of it. This is an issue. So first of all, you have to be, the self-management has to be there, and being able to also be reflective and not put your personal experience in front and your personal benefit in front. You're there as all researchers, like Simon is not putting so everyone is there to collaborate and getting better care and better treatment for cancer people or any other disease. So this is one of the issues that has to come first, and someone that is being to share his related experience to the related subject, instead of just having, well, I know that. I don't care about what you know. Tell me what you lived related to the subjects. This is me, but this is what I keep saying. Tell me how you are on that subject.
Yeah, thank you. And so I guess next question is, what advice might you have? And I turn to you, Simon, too, and Sandy, well, feel free to turn on your camera as well. So I guess turning that around for any maybe young researcher or a researcher that's looking to engage patients as a patient partner on their research. So noted about advocacy versus, maybe what we're talking about here today. What advice would you have for a researcher, for trainee?
Maybe I can start and Simon can keep going. There's two keywords that now for 6 years: willingness and readiness. If not one of the other is not there, don't start. Or it's because of COVID, Simon and Sandy were really willing, but COVID was not at a good time. So the readiness was not there. So they waited, but this is one of the two keywords. Simon, I don't know if you want to add that.
Yeah, well, it's patient engagement in research is something we all learned together at doing since a few years, and we have the chance here in Montreal to have this center led by Dr. Antoine Boivin, who's organized a structure that facilitates the involvement of patients into our research activities and collaboration. And truly, I think this, in centers where there is such resources, or it's really key to move forward, because as investigators, scientists, physicians, we're so focused in our training in the scientific aspects of things, and in the daily care of patients and complication and statistics, but we have never looked at the experience of the patients going through this. And I certainly have never thought of how to accompany a patient that is realizing that there's no standard of care anymore, and this crazy scientist comes with a very complex cell therapy approach and say, hey, this is great, and I'm sure you can do it. And what happens in the life of that patient is huge. And as an investigator in an academic structure with an early phase trial unit, and we're not ready to accompany patients into making the right decision, understanding. We're not prepared for that, and we don't have the structure that Sylvain Bédard and his team is able to provide. Specifically, in answer to your question, Megan, I think we have to team up with groups like Sylvain and Antoine's team, who are building this new way of supporting research and of supporting patients into research in specific context, which varies a lot. Chronic disease is not the same thing as last resort cancer treatment, like this treatment we're proposing. So teaming up with expert patient collaborators is key to move forward in this and to learn together how to best do it.
If I can add for young researchers, I just want to tell all young researchers that now CIHR is pushing a lot more on having patient, they call it patient with live experience or individuals with live experience on diabetes, cancer, heart failure. So there's the more we go, the more the experience, the input of patients' experience is empowered in research for multiple reasons, but this is coming up. And I worked with Kirk Scholl from CBMTG, was a president at the time. And he said, I like to talk to you because when I see you, I can ask you questions that my colleagues would not allow me to ask. So putting the input of the risk and benefit of factors that is still in statistic instead of human perspective or patient perspective, this is a great issue. And yes, we could be in basic science. I'm pretty good now in T cells, B cells with Mélanie Dieudet and Kurt and Jean-Sébastien. I mostly understand all of that. So don't underestimate the knowledge of the patient, and maybe use them to make yourself more understandable for your other colleagues that don't understand your language sometimes with T cells and B cells. Yeah.
Thanks both for that answer. So I'd like to pivot a little bit. So I know many years ago when your project got started, you were at the start of working through GMP, the aches and pains, and developing a clinical grade project. So very, very open-ended question, but any tips you wish you had known at the offset of this that you'd like to convey or discuss maybe more fully? Entered a little bit of an advice session for participants here.
Sandy, do you want to start?
Sure. I mean, the first tip would be to find the right people to help you develop a new project. I went from basic research to clinicals, so everything was new to me. So really, it's just ask the right questions and have the right people to answer your questions, I would say, is the most important, because you can't do everything by yourself, and you can't improvise to be a good GMP coordinator and then being a QC specialist and a QA specialist and a regulatory affairs specialist. You can't do everything by yourself. So that would be my advice.
Yeah. And on our end, we're in an institution where there has never been cell production for a trial. And so we decided to team up with the Center of Excellence in Cell Therapy attached to Maisonneuve-Rosemont Hospital, who had this academic GMP facility to produce cells. I think that was really wise to start in a first trial doing one step at our center, which is the selection of the T cells, but the other step, the expansion, is going to be done in a already up and running GMP facility. So this allows afterwards to transfer knowledge to our facility, and I think it made us fully realize what QA and QC specifically applied to cell production means. Because at the beginning, we were very happy to know that there were some QA and QC expertise at our center, but it was not applied to cell therapy, and it wasn't sufficient. And getting the institution also realized that earlier on would have helped us. And training ourselves be with specific training that BioCanRx sponsored on clinical trial application and on also Sandy attended many GMP training offered by the Stem Cell Network, CellCan, and in specific meetings there are some sessions for GMP. All this was crucial to open up the possibility of doing a cell product in our own hand and further develop new cell products for future trials.
Thank you. And I think what I'm hearing from this and all of our other sessions, right, is that it takes a village to bring a product to the phase of clinical trial and of course beyond. And of course, it involves different people with different expertise and different infrastructures and whatnot. So I think that's an important lesson. It's always nice to be mindful that you have to start early for many of these things because the relationships with your patient partners takes time, and securing the funds and the timelines and everything also takes time. So I think those are really important notes to make. So we have about 5 minutes left. I don't see any questions coming in, but with that being said, if there are any participants that are really yearning to ask another question, don't hesitate to write in the box. But I just wanted to, again, bring it back to Sylvain and talk a little bit about that and maybe training. So to give a little bit of background and as a promotional heads up to people who may be listening, to trainees in particular, we offer what we call the Learning Institute, which is an opportunity for patients or people interested in being involved in research to come to our annual scientific conference and be paired with a trainee to learn more about what's going on in the world of cancer immunotherapy. And so that's one mechanism that we're trying to train people who are patient partners who are interested in engaging in research. But so then I'm looking to you and I want to know maybe what training mechanisms there are either with your group or that you've seen as a best practice to help both researchers and potential patient partners have a strong relationship. Yeah.
So what we've done, I could say hundreds of times in the last 6 years, is we have a training session that are developed for researchers and patients. And the best of all is usually when you have a new project, we actually try to get the training one-on-one with the patient and the researchers to have, or the clinicians, to have the team together and having the different perspective on it. So this is what we've done. We've done it for SPORE, we've done it for CIHR. So this is a common thing that is just the basic of understanding what is the difference between patient partnership, patient advocacy, and patient subjects. So there's things like that, but also how to include patient, how to motivate patient. And usually after that, so we do that for most of every project. So the team is at the same level on knowing what is the patient involvement. And I'm actually doing at the CDTRP with Dr. Marie-Chantal Fortin, who's a nephrologist. We're actually measuring the impact of involving patient in research at the CDTRP. And one of the preview that we have in the statistic that we got is that actually clinicians are more afraid of collaborating with patient than patient collaborate with doctors. And I said, well, it's just normal because when you are in transplantation, you're used to deal with the doctor. It's like I will say a teamwork, and it becomes more or less your best friend because we're on that endeavor together because we're challenged, but let's try to win that game. So coming back to the Canadians, but so this is and then the researchers or the clinicians are afraid that it's just a limit that they don't know where so there's a lot of ethical concerns. But once you clarify the objective, what are the limits, blah, 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 confidentiality, it's all good. And this is all in the coaching sessions that we have.
Kudos, that sounds like a fantastic program. So congratulations to you and Dr. Beauvais and everyone who's involved. So that brings us to 3 o'clock. I'd just like to end by saying thanks everyone for participating. That was a really comprehensive session and I appreciate your time, and to the participants as well for their questions. If you only caught part, or if you want to watch this again, the video will be posted on cancersummit.ca, and so stay tuned for that in the next couple of days. And as always, follow us on social media to be informed of upcoming events. So with that being said, thanks everyone, and I guess go Habs go. I'm also not a fan necessarily, but go Habs go. All right, thank you everyone.
Thank you.
Thank you. Bye.
Thanks a
Bye
lot.
now.
Merci, bye.